right, this little portion, we're going to talk a little bit about diagnostics. And and what you see on the screen is just a definition straight out of an English dictionary, saying that the diagnosis is an act or an art of identifying disease from its signs and symptoms. This is just a general definition. The one thing that I do want to point out, anyone can do a plant disease diagnosis. It's an art. Plant pathology is a science. So we use knowledge that we gain from the science to be able to make sense, and how we use that knowledge to come up with answers is an art. It's a skill that can be learned. Anyone can do this, and with practice, you can get better at it. And so that should be encouragement to every one of you that plant disease diagnostics is not difficult. It may be cumbersome at times, but you can always get better at it. Now, let me also make a statement. All of you on this Zoom have made a plant health diagnosis before. And I will even go as far to say that every one of us on this Zoom meeting has not only made a plant health diagnosis, but also has made recommendations before. And if you go around and said, well, I don't think I did that. Well, if you actually think back and you actually ever pointed or looked at a plant and said, that plant needs water, well, you have made a diagnosis and you have made a recommendation. The diagnosis was that you assume, based on certain observations, that the plant lacked water, or basically were exhibiting drought-like symptoms. And your remedy for that diagnosis was to add water. A lot of folks have done that. Is it a right diagnosis? Well, great question, which is what I'm going to share with you next, is this thing, what I call a diagnostic continuum. It's not a one-size-fits-all. So several years ago, I was asked to write a chapter on diagnostics for a textbook, and I drew a line chart. So this is infinitely much better looking than what is in the textbook. But I wanted to point out that on the left side, we have this thing called simple diagnostics. And what I want to show is, for the most part, many of us have done or practiced simple diagnostics, or what we call field diagnostics. Master gardeners are more likely to basically do field diagnostics or simple diagnostics. Basically what it is we're taking observations, we're using prior knowledge, we're making comparisons with those prior knowledge of known symptoms, and making a conclusion based on comparisons of what we see of a current subject or patient against those known reported symptoms. So it's experiential to a degree. So to give you a very simple example, let's just say Jayla calls me up because she realizes I'm the plant pathologist and said, hey, my yard, I got this little brown spots in the lawn. What is it? You know, me being the smart guy I am, I heard brown, I heard spots, and I tell her, I said, hey, you know, you got brown patch on your lawn. Well, the thing is, how accurate was my diagnosis? And hopefully you guys would look at that and said, wait a minute. We don't know what type of grass she had. We don't know how big those spots were. We don't know when those spots appear. We don't even know if she has a dog, or we don't know what type of influenza was there. There could be a ton of questions that I could have asked to basically make that a better diagnosis. But nonetheless, my response was, in fact, a diagnosis, a very simple one, a very simplistic approach based on a small piece of information. Simple diagnostics, that is the extreme. Now, on the other hand, or the other end of the spectrum, is what we call research. What happens when you get the question is, I want to know for sure that this organism is in fact the pathogen. Show me proof. And we call that proof of pathogenicity. And there is a procedure that was named for a German scientist. It's called Koch's postulate. And this Koch's postulate was actually first developed for animal diseases, of all things, anthrax. Uh, and and basically, the concept which we do in plant pathology is it takes this approach. You have a plant that is sick, you should be able to describe the symptoms on it, and you should be able to isolate an alleged pathogen from that sick plant, and you should be able to identify that alleged pathogen. Once you have the identity of that alleged pathogen, you take that alleged pathogen and introduce it back to a healthy plant. So, in other words, you use what you isolated, you identified it, and you inoculate a new plant. You should see symptoms that are similar showing up on that inoculated plant. And then you should be able to re-isolate that microorganism that you inoculated onto that plant and demonstrate that it is identical to what you use or to inoculate that plant. And when you have done all those steps, you have what is known as satisfied Koch's postulate, shown proof of pathogenicity. One key thing here, how long do you think that takes? Well, it really depends on the pathogen. If you deal with a bacterial pathogen that multiplies quickly, that may be, a research project that takes weeks. If you deal with a fungal pathogen, depends on the life cycle of that fungal pathogen. It could take months. And in the case of like cedar apple rust, I told you it was a 2 year to 3 year life cycle. That may be a 2 year, 3 year study easily. I actually had a student that published a first report where she did Koch's postulate on a particular fungal agent of boxwood. And we had, everything was set up for her in terms of we knew the identity of the pathogen that we isolated, and all she had to do was inoculate the plant and then re-isolate the pathogen. What was planned for 10 weeks ended up taking somewhere about 16 to 18 weeks. So, when you deal with biological systems, there's always potential for things that does not stay exactly to timeline. And the fact that when you're dealing with a research project, it does take a lot of time. So in a diagnostic clinic like the one I run, people want to know answers. They don't want to wait weeks or years for answers. So how do we deal with what we call a happy medium? And so what we have here is this thing called confirmatory diagnosis. So if you see that red arrow there in the middle, confirmatory diagnostics take advantage of the research that has been done, because invariably through that research that has been done, there would be detection methods that have been developed. So we would have tools that we can use to identify certain organisms. But it also takes advantage the experience of a diagnostician. So when a sample comes into a diagnostic clinic, to the ER for that matter, what happens is a diagnostician will have to look at it and see those visible symptoms and said, wow, that could be caused by X, Y, or Z. And if there are tests for X, Y, or Z, she might write, or he might write an order to say, take the sample and do a test to see if X is present or Y is present. Now, keeping that in mind, what happens if the test comes back and said X is present? Then the diagnostician has to make the decision and say, I see symptoms that are consistent with what X could produce. And now I have proof that X is present. Therefore, by the virtue of those information, I am going to make the decision that this plant is infected by agent X. Now, the point here is this: it's what I call indirect proof. You have proof that the pathogen is present, but you don't have proof that the pathogen actually caused disease. What you do have is damage that looks like it could be done by agent X. So that's what confirmatory diagnosis is. So taking tools that have been developed through the research project and utilizing it to identify the presence or absence of a pathogen. And by doing that, you save quite a bit of time. So now a diagnostic could take days, could take weeks instead of many months or years. So that is the idea of confirmatory diagnostics. And when you look at this diagnostic continuum, these are the three things I want you to remember. When you go from left to right, from simple diagnostics, which most of us do, to research, there's an increase in complexity of method. Simple diagnostics, just information that you get. And obviously, you want a better diagnosis. You ask more questions. When you ask more questions, that it gets a little bit more complex. That's the way it is. And when you ask more questions too, it takes a little bit more time to engage. And so, or if you send it to a lab to do a test, that takes a lot of time to engage, more time than just asking questions alone. So when you go from left to right, you do see an increase in the amount of time that it would take to come up with an answer. But on the same token, when you go from left to right, not only does it increase in time to come up with an answer, or the complexity of the method that's used to try and get that answer increases, you also increase your confidence in that answer. So more proof. You can perhaps put a little bit more credence in the answer that you get in trying to understand whether a certain pathogen is in fact the disease-causing agent. So that is kind of the concept of a diagnostic continuum, to put it in context in your mind. Most of what you're going to do as a master gardener is going to fall on the area of simple or field diagnostics. Asking the right questions is going to help tremendously. Experience is going to help with your ability to do good diagnostics along the way. So those are all skill sets that can be learned and can be gained. Now, I want to go quickly talk about practical diagnostics in terms of just to give you a tool to help you how to do diagnostics. And by the way, this is one of many approaches to doing diagnostics. When you do diagnostics, it's really coming up with a sensible, reasonable, and effective method of maintaining information to help you make decisions. And so what you see on the screen is what I call the Ong method of practical diagnostics. It's just a simple guide about how I do things. It may work for you, it may not work for you, but ultimately, when you do diagnostics, think about what are the ways that you can gather information and use it in a logical sequence to help you to get to an answer. So let me share with you my 5-step method. I call it 5-step because you can fit it into one hand. Number one, identify the plant. If at all possible, is it always necessary to identify the plant down to the species level? No, it's not. Would it be helpful if you did? Absolutely, it would. If at all possible, should you identify the plant? Yes. Not to kill yourself if you don't know what it is, but even if you get the right family, it's going to be helpful to have that sort of information. One of the reasons is if you know what type of plant it is, then you can very quickly tell whether that plant is how that plant is supposed to act and look at that particular time of the year. You guys might not realize this, but every year around this time, starting in March, going through about May, we get phone calls and some master gardener groups, like I know Dallas County gets phone calls around April or so, where folks would call up in a panic. You got to help me. My oak trees are dying because it's losing leaves. Well, then you start asking questions to find out what kind of oak tree it is. And if they don't know what it is, and you start asking some probing questions and you realize they're referring to a live oak, some of you might realize, wait a minute, live oaks in Texas around March, April, they start losing their leaves naturally. It's a spring leaf drop. So that's really not a disease problem. That's not a problem in that part. So when you identify the plant, you can quickly tell what is natural and what's not. And if you think back about the definition of a plant disease, is something that's abnormal happening to the plant. If it's normal, problem solved. If it's not normal, then you proceed on to two, which is assess the nature of the problem. Well, okay, we know something is abnormal. So what's the issue? And when you assess the nature of the problem, is sort of like taking a historical medical record. You know, when you go to the medical doctor, if you go to a new doctor, they have you fill out this ton of forms, checklists of what kind of problems you might have in the past and so on. And in fact, now I think they make you do it annually. So I'll tell you one thing. I go to my family physician one year. They had me fill this out, and I go and I said, and I told my GP, I said, do you actually read the darn thing? He goes, yeah, I do. And then I started quizzing him, and he does read that darn thing. I said, why do you read that thing? And he's a smart aleck guy like me too. He says, same reason why you get information when you do your diagnosis, figure out what's the background. And so when we talk about assessing the nature of the problem, information for those things are sort of like, when was the plant planted? When did you first see the damage on the plant that you are having a problem with? What are some of the environmental conditions that happened in the week prior or 2 weeks prior to the symptoms showing up? All those pieces of information can be helpful in coming up with a list of suspects, which is point number 3. You know, so as you gather information to assess the nature of the problem, invariably you will come up with a list of suspects. And as you continue to gather that information, you get to a point where the more information you get, it starts kicking out some of your list of suspects, because you just said, wait, it's not possible that this particular suspect is present because of this situation. So you begin to narrow your list of suspects. That's point number 4. And hopefully, if this is done correctly, you would get down to one answer. The likelihood of that happening usually is very small. You probably end up with a list of 2, 3, 4, sometimes 5. But what happens here when you get to stage 4, when you narrow the list through that natural process of collecting information and coming up with the list of suspects and subsequently eliminating suspects and narrowing it down to several, you can take those several suspects that were left on the table and look for information, research that has been done or what is known by others about those particular pathogens and the disease they cause. And you might find little nuances in there which will help you differentiate one from the other. I'll give you an example. In Texas, we on oak trees, we do have 2 leaf spot diseases that occur in the summer. That would be anthracnose, which is caused by Colitotricum or Gliosporium sporoides, a fungus. And we have Tubacia leaf spot, which or formerly known as Actinopelty leaf spot, caused by the fungus Tubacia. Both of them cause a spot on leaves. It happens on both live oaks, but also on red oaks, particularly bad on red oaks. Usually happen in the late part of summers when there is some moisture involved. Well, when you look at their profile and you go through the assessment of the problem, what you find is they show up around the same time of the year. They cause leaf spots. They basically cause damage with you and respond to influences that are quite similar. So you end up through that natural process of elimination and end up with two of these probabilities. You go look at literature, you go look at research, And you will notice that it will say that Calototricum ogliosporoides basically have preference when there is increased humidity compared with Tubacia leaf spot. So then if you start thinking about that for a minute and you read between the lines and you go back, look at your notes to see if your notes, you ever made a notation, whether the problem started within the canopy of the tree and spreading out, or did it happen on the outside of the canopy and not really in the middle of the canopy? Well, what's the difference? Well, if the complaint was that you had spots happening, what looks like defoliation in the middle of the canopy and moving out, gee, hopefully you should be able to say that, oh, that is most likely Calototricum anthracnose. Why? Because within the canopy itself is a microenvironment where it captures the humidity, so the humidity tends to be higher. So that would be a preferential environment or influencer for anthracnose rather than Tubacia. So little things like that, when you get to point number 4 and you end up with 2 on the elimination list, you can go and look for previous experiences, previous research that has been done to get those little nuggets to help you determine whether it's one or the other. So that's how you would use something like this. Now, I didn't go through very much on practical diagnostics. We do have a program with the what we call Master Garner Advanced Training First Detector that we don't really do a plant pathology diagnostic training, in the sense that it's often very difficult to have samples ready to look at. But the First Detector program is more of an outreach awareness program, trying to train Master Garner volunteers in areas to do what we call surveillance, and to be prepared to know how to read some of those literature, and so that they can be extra eyes and hands for the lookout of things that are important in terms of pests and pathogen to this state and to this country. So previous programs that look at disease models such as rose rosette or citrus greening or citrus canker, some of the palm phytoplasma diseases. And one year, I believe we did some things that did with emerald ash borer, and one year with brown marmorated stink bug. So if you notice here, it's just not pathogen alone, but pests. And I'm hoping that this year we'll be able to run one off virtually, but that still remains to be seen how we proceed with that. Now, for the next few minutes, I want to plug the department that I'm in. So the Department of Plant Pathology and Microbiology at Texas A&M University, we have a new building that we moved into a little over a year ago. Well, about a year and a half ago, which is the picture that you see. It's next to the horticulture building, if you're familiar at all with the campus. But we have moved from the main campus to West Campus. So we're in one of the newer buildings. What's interesting about this building, when it was built, the research wing, which is facing you in that picture, is actually a certified USDA biocontainment facility. In other words, there are checks and balances in place and equipment in place to basically create a containment environment in there, so that we can study some pathogens that are perhaps disliked by horticulturists and most folks. The Department of Plant Pathology and Microbiology, we do have an undergraduate program called Bioenvironmental Sciences. And you might think, wait a minute, how is it that a plant pathology and microbiology program would have something that is bioenvironmental? Quite easily, because the part of the microbiology, we see a lot of organisms that have a role not only in plants, but how it affects the environment and how the environment itself affects the microorganisms. So it's a relationship, things that's occurring. So the undergraduate program actually bore out of one of the interests to provide undergraduate students with a broad-based science background to allow them to pursue further things that would involve environment and things that would involve microorganisms. And you might be surprised that some of our graduates move on. We got quite a number of our graduates that move on to a degree in public health, whether it's policy or practice. We have a number of students that actually end up in law school and doing environmental law. In fact, one of my first undergraduate students 20 years ago, she ended up going to law school, and now she's still in the Marines as a Marine lawyer, JAG, doing environmental law. So we have a number of folks that did that. We do have a number of graduates that basically took that knowledge and go into the waste management industry. And so they make their buck that way. We have some folks that actually probably have good bedside manners and take that knowledge and go on to medical school or vet school, less likely vet school. And we got a few, unfortunately, too few, that actually decide to pursue things further in plants itself. But there's a lot of areas of interest in the bioenvironment. In fact, the two latest hire in research that we have, one is actually in the area of molecular epidemiology, trying to understand how things move in this world. And the latest hire that we have, Dr. Anthony Babu, is what we call a population microbiologist. He's looking at how microorganisms interact with each other in order to protect a plant. And one of his fun projects that he's doing is he's looking to see how we can modify soil environments to make it conducive for truffle production in Texas. You might say, what does that has to do with pathogen? Well, nothing much really, but the fact that some of those truffles are fungi or fungal agents that interact with plants in some way. It's not a parasitic advantage, but it's a symbiotic relationship. So the question is, how do we get those organisms to communicate with each other, to find a place to be able to create, let's say, a pecan orchard into a truffle farm? He's nowhere close to an answer yet, but those are the sort of type of questions that we ask and we explore. On my part, who's a straight plant pathologist, on my research part, we do a lot of work on detection technologies. I'm working right now on rosette disease is one of them, but another one is the virus in tomatoes that officially does not exist in the United States. And it's under quarantine right now that we have federal orders that limit the import of tomatoes and peppers from Canada and tomato and other countries into the United States because of this particular virus. And so I do work with not only with us here with USDA, but also with the Canadians and Mexicans in order to help mitigate some of those type of issues. So there's a lot of fun things going on. And I'm going to extend this invitation to you guys. We are next to the Hort building. We're not too far from the gardens. Hopefully when things actually improve in this pandemic era and you're thinking about an outing for your Master Gardener group, do consider us at the clinic and the Department of Plant Pathology. You're most welcome to come and visit the Plant Pathology building. And the gardens is on the way. The plant clinic is about 3/4 of a mile away, all within walking distance, which would make a nice tour to actually look at the resources that are available at Texas A&M that Master Gardeners can utilize. Oh, I also forgot to mention, you still have the Insect Museum over at Heat Building, just a stone throw away from the Plant Path building. And so you have all these resources here that's available. Um, so so if you got kids, grandkids, you want them to have a good science background, send them our way. That's called recruiting, by the way. So I did my part recruiting for the department. Let's talk a little bit about the plant clinic. The plant clinic, we are part of the National Plant Diagnostic Network. Now, the Plant Diagnostic Network was the result of a response to potential bioterrorism. So I think most of you may still remember 9/11/2001. We had a terrorist attack. And post that terrorist attack, what happened was we had a number of folks that was skittish and came up with a list of something like 108 microorganisms that could be used as a bioterrorist agent. On that 108, most of them were animal and human pathogens, but 10 of those at that time were plant pathogens. And this was this is called the select agent list. And so the question came up in Congress was, well, how do we monitor human diseases? Well, most of you know that answer because we see the Center for Disease Control in the news a lot these days. And then the question also came out, how do we monitor animal diseases and how do we monitor plant health or plant disease issues? Well, there was no answer for the animals or the plants side of things. So in 2002, there was established the Agricultural Bioterrorism Act, or there's actually another name for this particular act that was established in 2002 that set aside some money to create new networks. The National Plant Diagnostic Network was one of those networks that basically connected all the diagnostic labs, existing ones, usually associated with land-grant university, like Texas A&M in Texas, so that we can better communicate with each other. Or it may tie in some diagnostic labs from State Department of Agriculture. In states that do not have those diagnostic labs, they did provide dollars to establish those labs. So this was in 2002. There was also a network that was set up for animals. That's the National Animal Health Laboratory Network, which A&M, the TVMDL, was a result of that part where Texas was one of the hubs for the animal science and animal health network. Now, specifically for the National Plant Diagnostic Network, what the network does is it focuses specifically on plant problems with the idea that it's a way to rapidly detect, diagnose, and disseminate information specifically on high consequence plant pathogen, insects, weeds. So it's just not pathogens, but it includes insect, weeds, and other biological pests. So this network basically cares for plant health. Now, to administer something that large back in 2000, they actually split it into regions. And so there were 5 regions. This was in 2002 when they split how they would divvy up the dollars. And so if you notice something about Texas, you notice Texas was kind of split in 2. So we do have some very influential people in Congress now, and we also had some influential people in Congress then, that because Texas was so large in terms of the type of crops that we did, but also with the type of institutions that we had, Texas essentially got double dose, so to speak. The clinic in College Station has been around since 1957, so it's an existing clinic that got folded in. But I guess they decide to give some dollars to Texas Tech to start a diagnostic clinic for the north part of Texas. Well, what happened was, I think Texas Tech realized that the money was not sufficient to actually start up a bona fide clinic. So that money got bumped around and ended up with AgriLife. And as a result, AgriLife Extension got that dollars and set up a clinic in 2005 in Amarillo that was just functioning as a presence. And in 2008, we hired, not 2008, what year is this? 2018. we actually hired uh, right, we actually hired a person to run a clinic up at the in Amarillo. And so we do have two clinics in as a result of that funding in the state of Texas. Well, a couple of years ago, they decided, when I say they, the network decided they didn't want to deal with multiple contracts in a state. So each state now will only have one contract. And as a result, um, Texas is down to one contract. And we actually, well, I had to deal with a cut in budget to run two clinics. And so that's what happened this year. We do have two clinics, but with less federal funding involved. And so we actually find some other sources of funding, whether through state or through other federal programs. Um, so that's the way that the National Plant Diagnostic Network look right now. And as of current, we have a third NPDN lab in Weslaco, which is the virus lab that I mentioned to you about. So under the umbrella of AgriLife Extension and the Department of Plant Pathology and Microbiology, we operate three diagnostic units in this state. Keep in mind that we do, these are fee-based labs because we have to do cost recovery. Doing diagnostics using tools and all that is not cheap. And so in a diagnostic clinic like the one that we have in College Station, our base fee is $35 for in-state. We do have a number of folks that utilize us from out of state where we tag on a $20 surcharge because our goal for our state is to take care of our people first. Just to show you a few things that we do at the plant clinic, everything from traditional approaches to diagnostics, which is culturing and microscopy. So the lady that's in the pink lab coat there, that's Sheila McBride, who is my program specialist and head diagnostician. She does some of the master gardener training as well for certain counties. And if you're in Denton County, I think she's scheduled to do one on trees for you guys coming up real soon, or if that not already happened. So in a clinic like ours, we actually teach or have the opportunity to engage students and give them exposure to do the traditional arts of diagnostics, which is microscopy and culturing. Those 4 plates of Petri dishes that you see down here is our test for oak wilt. We also do molecular work. So the newer tools that might use a PCR machine and running a gel. This is an example of one of those. So a clinic is very unique in a sense. The service clinic is not like a research lab. We have a lot of different pieces of equipment because we have to be ready to respond to any sort of things that is thrown our way to be able to make those decisions to help identify what the problem is. You might ask the question, are you guys always accurate and in fact correct? So back in the 1980s, there was a survey that was done among diagnostic clinics at Land-Grant University, basically just asking diagnosticians on their confidence level. And surprisingly, that confidence levels say that, diagnosticians feel very confident of their diagnostics about 48% of the time. What it is we are trying to make educated guesses. Now, I'm going to say this, diagnostician says that it's 48%, but the percentage is infinitely, I believe, much higher, because I think a lot of times diagnostician tends to doubt themselves. But what we try to do is find evidences of the presence of the pathogen to be able to make that call. Are we 100% accurate? No. But based on the evidence that we have and the sample that we have, the response or the results that we provide are based on that sample that we have in our hands and what we can see and what we can isolate. At that time, that sample is within our possession. Now, the Plant Clinic do have a presence online. the one thing that I do want to point out is our Facebook site, where my folks are pretty active. If you have not found it yet, please go look for it. You'll see the tag here at the bottom, TX Plant Clinic. My staff will usually post a What Wednesday video with some topic area that is interesting for folks. So go check it out. I usually put a Friday post or weekend post on something that's happening around my neck of the woods. So please go check that out. Like us and follow us. Now, let me talk a little bit about good samples at the clinic. It's one of the pet peeves of diagnosticians, but at the same time, there's a lot of things that you can learn about what would constitute a good or bad sample. And even with simple diagnostics, this knowledge is going to help you to identify what sort of samples might be usable or would be infinitely usable in terms of trying to make a decision as to what's going on. So at the Plant Diagnostic Lab, the one thing that I want to remind folks is diagnosis is really dependent on the quality of the submitted sample. So the only thing in the clinic that we see is we don't see the field, we don't see your backyard. What we have is the plant, the sick plant. We can try our best to get some information off the form regarding where the plant was and some of the history. But the fact of the matter is all we see is the plant that we have at hand. So the quality of that sample itself is very important in order to return a good diagnosis. So these are some of the do's that we suggest to folks. And this is something you can do too when you work with clientele in your counties. Ideally, start with photos or a call for support. If you don't know what you're looking at or you don't know what sample you ought to be taking, take pictures of what you think is wrong. Make that phone call to say, what would be an appropriate sample to submit? The clinic itself, we have folks that would be glad to look at that and provide some suggestions. In a county type situation, you might get an email or phone call. It's always good to say, hey, could you take a picture of what you think is wrong or what you see is wrong on your plant and email that to us. Pictures is worth 1,000 words. Even then, we also want to keep in mind that when you do take pictures, you want to make sure that you do have things in focus. And nowadays, most of us have a camera in our pockets or in our possession, with those new smartphones and all that. And remember, when you take a picture, you can always look at it and know whether it's in focus or not before you hit send. So do that. Take samples prior to any treatment. This is a pet peeve of the clinic because one of the things is we deal with a lot of producers and so, or even landscape maintenance folks. And what they do is they go out there and they look at a plant and they realize the plant is not well. And so they treat that whole area. And then they decide they want to know what's wrong with the plant. And so they take a sample and send it to us. Here lies the problem. If they chose correctly or they use a broad spectrum, that product could actually interfere with the growth of the pathogen. So when it comes to confirmatory diagnosis, where we try to detect it, where we try to recover it, we might fail because of the interaction of that organism with a product that may have been used. So we advise folks, we said, if you think something is wrong, pull a sample before you have to treat it. And in fact, that's how we talk about green professionals using the plant clinic is for confirmatory type situations. You don't have time to respond. You might have to respond to that client that same day. Well, go ahead and respond, but pull a sample and send that to get confirmation because hopefully in about a week or 2 weeks when you get an answer back, you either confirm what you saw or you learned something new. And if your prior treatment did not work, now you actually have an answer to perhaps select for a treatment that might work. And that's how a green professional should utilize a clinic, is for confirmatory purposes and for information that could be used into the future. Fresh samples with enough information. So one thing, when you submit to any clinic, there's always an intake form, a submission form. And we want samples as fresh as can be, because if you're dealing with biological agents, you want them fresh so that you're not fighting with other organisms to try and isolate them, or hopefully the sample have not been exposed to some harsh conditions that would affect the microorganism itself. When I first started, we had some issues with some county agents in agronomic fields, where they would go out on field visits, and they would take a sample and they would throw that sample on their dashboard. And then 2 days later, they would put it in the mail and send it to me. And I would call them up and said, there's nothing wrong with this. I don't see any pathogen. They were like, well, we know we had it. The leaves was symptomatic when I took the sample. Well, when you send it to me, there was no leaves. What happened? Oh, it dropped. Well, where did you put the sample? On my dashboard. Were you out in the field the other 2 days? Yeah. In the summer on the dashboard, that's cooking the sample. And if you do cook the sample, you're going to cook anything that's alive in it as well. So it's not a surprise that when you treat a sample that way and it gets to us, you're going to get a no pathogen found result. So fresh samples is very important. As much information that you think might be helpful to the diagnosis is also going to be important. The last part, where I talk about chain of custody, we deal with some regulatory samples. So whenever you deal with regulatory samples, you do want a good chain of custody. And this is true with any samples. We want to be able to say that when we get the sample, the last person touched the sample was either the person submitting it or whoever is recorded as the sample. In other words, we want to be able to say that this sample was not tampered with from the time it was sent to us. So then we can actually try and keep the integrity of that sample. A question that's just popped up in the chat is, do we provide guidance for packaging sample? Yes, we do. And in fact, there are a few slides that I'll talk about it, but we actually have 1 or 2 videos actually on our YouTube site that you can actually go check that out. And in fact, we had a What Wednesday video on that. And not only that, we do have a What Wednesday video taken from the viewpoint of the sample itself going through the clinic. So those are things that if you have time, you can check out on our YouTube channel. The question often that we get is, what is a sufficient sample? Now, if you ask a diagnostician that, they would say the whole plant would be best because we can actually see the whole thing. Well, I think you realize very quickly that it's not true for all types of plants. We have limitations, especially in terms of space, and it also depended on the plant type. If this was a bedding plant, a transplant, sure, the whole plant itself, like that image on that right, it's great because we get that whole plant that we can actually do a full analysis on the whole plant. But when you get a plant in this situation, this was a 1.5 inch width. That's about 8 feet tall. Okay, it takes up a lot of space, but it still gives us an opportunity to look at it. Do we want a lot of this sort of samples in the clinic? No, we don't. So how do you deal with a situation where somebody might want to send in a sample of a tree, an established tree? Well, what we say is get a representative sample. In some cases, that might be a branch. In some cases, it might be the branch and the root, or part of the bark or part of the branch, to try and provide an image to the diagnostician of what this might look like in nature. And so by doing that, it gives the diagnostician the sample, the knowledge, the information that's needed to make a good decision to help determine what has gone wrong. Now, in terms of a good sample here, I'm going to just point out in terms of turf, because this is a nice visual here. This person sent in 3 cups of turf grass sample. And where I pointed down on the in the field, in the sort farm, you have one that came from what looks like the healthy part, one that came from where it was all dead, so where it's severe, and one came from the transition. And in fact, when you look at it, you could probably make out the line where this part is the dead part and this part is the life part. When a sample like this comes into the clinic, my technicians and my diagnostician will gravitate to this particular sample first, the transition sample, because the theory is always this: when you have transition, that is where a pathogen, if a pathogen is present, is most likely to be active. And if it's active, we can probably recover it and isolate it. The usefulness of the other two things, or the other two samples there, is this. Once you can identify what the problem is, you look back on a sample that came from a part that's all dead. And there may be an opportunity there to do some forensics to confirm that the damage, and perhaps may even find the pathogen itself that is contributing to that active parasitization. A sample from the healthy area will allow us to look at it to see if there's any evidence of the pathogens that was found in the other two. Because if we do see them here, at usually at low levels, we can inform the submitter that when they treat it, they would have to treat even the healthy areas, because we have evidence that the culprit are also found in the healthy area, perhaps not at the level to cause damage yet. So this is an example of how a sampling of an area that shows a good representation of the damaged area provide information to help the diagnosis and the recommendation. Some of the things that we put, and one thing I do want to point out, the last thing is we do say send overnight in a crush-proof box. Guidance here: if you send a whole plant, realize the roots are usually in some form of media or soil. There's a lot of microbes in the soil. So we would always say, if you can isolate those roots or the parts, so that when during shipping, what you don't want to happen is soil particles that get on the foliage, and that can contaminate those foliage, creating opportune areas for other microbes to colonize, and as such could interfere with recovery and identification of potential pathogens. So an example here is like, take a plastic bag, wrap up the root ball and just tie it around, basically isolating that root ball and any of the soil from causing contamination on the foliage. When you put it in a bag, some people think, oh my gosh, if you're dealing with biologicals, we want to put in wet paper towels. No, you don't. There's usually enough moisture in there. So if you want to put any paper towels at all, dry paper towels would be great. If you create too moist of a condition, sometimes you can encourage a lot of the saprophytes or the opportunistic organisms to grow, and they can actually overcome or overrun the pathogen itself. That's going to make the diagnostic process a little bit more difficult at the clinic level. Earlier, I mentioned putting this in a crush-proof box. So this was actually images from the North Carolina Diagnostic Lab that my colleagues shared with me. And we do get this at the Plant Clinic here in College Station as well, where we would get manila envelopes where the submitter has good intentions and write in big letters, said, please do not crush or fragile. And invariably inside, we would get samples that ended up like this, totally rotted, and potentially forms that are not only wet and sticky, but also smells. So in a situation like this, we say, crush-proof box, because you want to keep the structural integrity of the sample itself. If you put it in an envelope, even if it's bubble wrap, that mashing and all that stuff can create damage to the plant cells, releasing some of those nutrients from the plant cells, which can be a highly nutritious environment for other microorganisms to grow on it, which can disrupt or interfere with the diagnostic process. So to recap, in terms of samples, lessons that we know at the diagnostic clinic that may also help you is the diagnosis is only as good as a sample. So when you get a sample that somebody bring to you or show you a picture, realize your answer is only as good as what you can see and the information that you have. And the sample is only a representative of the situation of a larger context. You can make some guesses, but ultimately you don't know what the larger context is unless you have the opportunity to be in that larger context, observing that larger context. The amount of time that it takes to do a good diagnosis relates to the accuracy and confidence of the results. If it's something that is unique, something that is not known, oftentimes it may take us a month or a month and a half to get an answer. So there is, this, unfortunately, we don't have tools right now where we are able to turn around with an answer in 30 minutes or an hour or a day. It does take time for some of the diagnosis. Now, in terms of diseases, for simple diagnostics and then recommendations, I do want to point out that the Extension Plant Pathology group do have a resource that's available to you guys, which are extension fact sheets. I believe you can access this at the AgriLife bookstore. To access the Extension Plant Pathology series, just search for EPLP and it should bring up a bunch of different fact sheets. I believe we have a set for fruit trees. We do have a set for roses. We do have some, for example, for vegetables like basil downy mildew. I think we have one on carrot. So take a look at that. Most of this are going to be one pagers front and back, so that you can actually print out and share with your clients in that fashion. Now, the one thing that I'm not going to go through in detail, but to kind of introduce you to it, is what we call disease control principles. So when we do a full day training in plant pathology, we would capture solutions. So I would actually, and this is not a new idea. In fact, this came up through writings of a plant pathologist back in 1932 or 1934, Dr. H. Wetzel, that calls this control principles, where he took every potential solution and categorized them into certain groups. Now, over the years, people have basically modified the groups and utilize different terminologies to fit in those new technologies in terms of tools that are able to manage plant health. But there are a few things on this list that has remained true from 1934. For example, resistance. All that is referring to the innate ability of the plant to resist a particular pathogen. And so this is a genetics, a tool that we can use in terms of utilizing certain varieties that have resistance to avoid or to basically circumvent, to deal with the plant health issue. Other categories in this, what we call disease control principles, would be things like exclusion, protection, eradication, avoidance, and therapy. And just quickly go to exclusion. Exclusionary methods or strategies within here does apply at the home level all the way to the international level. And so when you think about plant protection and exclusion principles, what we have is quarantines that are in place. Because why? The federal government does not want certain things to come into and potentially affect our natural resources and our cropping systems if we don't have it in the United States. So those are sort of quarantines that can be imposed at the federal level as well as the state level. If you are a person that want that pristine garden and in your sovereignty of your home, you can impose a home order that any new plants that come into your garden has to pass inspection to be free from root damage. And what I mean by that, if you're concerned about having that pristine garden, one of the simple approaches that you can take is when you buy plants is to ensure that you have good roots. Something as simple as popping out that plant out of that pot and look at the roots. If the roots have brown or looks mushy, avoid those plants. It may be a natural occurrence, or it may have some kind of pathogen on it. So you don't want to introduce any potential pathogens into your garden. So in that way, you are practicing a strategy in the principle of exclusion. So the rest of this stuff, there are various things that you can stick into one of these principles, and basically it would capture all potential solutions that could be used in a plant health management program. So uh, with that, the part that I want to go over on the concepts, just to quickly recap, the two very important things that if you guys say, what do I need to remember from today? The two most important thing is the disease triangle and the disease cycle concept. If you understand those well, especially the disease cycle concept, regardless of where you are in this world, when you get information about disease, you should be able to learn or know the pathogen better and how it causes disease. And if you know how it actually lives and causes disease, you can understand better what would work or what would not work in terms of mitigating the damage that's caused by that pathogen. So the disease cycle concept is really important. We did go over a little bit of diagnostics and also went through very quickly on the disease control principles. Realized that those two things are just customary introduction. There are opportunities in the future for training to get more on training on diagnostics through either surveillance or skill development. And there's also potential for training where we talk about solutions, focusing particularly on different groups of disease type situations or incidences. But hopefully, what I've imparted for you today is to give you some resources, to give you some background foundational information that you can take and build upon. And this is what I call a start to the learning process. If you're interested in plant diseases, plant pathogens, there is a plethora of information out there. And there's a lot of interesting information that you can learn from it. And also realize one thing: pathogens are not necessarily bad or ugly. Some of the damage that they cause can actually be quite pretty. It depends on how you look at it. And sometimes the pathogen itself can be turned around and utilized as a tool to help to advance mankind. So keep that in mind, is we do have those type of things that are out there that can be learned, that can be read about, and all those can contribute to your ability to be able to determine what perhaps might be going wrong in terms of the health of a plant, and perhaps even encourage you to gain the skills and improve on the skills that you might need to do good diagnostics. On this slide here, I want to mention that I did mention to you a Facebook site, but we are also active on Twitter. We do cross-post it on Instagram. So if you have any one of those, just check us out, TX Plant Clinic. And hopefully you'll get a chance to look at that.